Okay, let's get started. What's happening on Wednesday? Midterm, yes. Which of the scantrons are you bringing? The big blue ones. So that's the first thing. The second one is I could have sworn I put clicker questions into here, but then again, they seem to have disappeared. Oh, shucks. The third thing, which I think is rather important that actually has absolutely nothing to do with this class, is that I'm an immigrant. Both my parents are immigrants, and I'm married to an immigrant. I had a green card for almost 30 years. I'm now a U.S. citizen, and I try and keep up with all of the rights and responsibilities that go with that. So, done, finished. Let's move on. So today, as usual, what I do with these, and I've been doing for the last 15 years, has to do with going through all the stuff that I think I did a really bad job of explaining before, or things that I think really need to be emphasized. Where the stuff's on the exam, I don't know. Is it done yet? No. I've still got like 48 hours, right? Like you're all done studying, right? Exactly. Same kind of deal. There was a quick question, sorry. Did you have your hand up?
I was going to ask you, where are you from?
Oh, originally from England. But my parents came here purely temporarily on temporary visas about 50 years ago. So, yeah. Rewind. Yeah, so the whole idea here really of this, tree of life, thinking about molecular biology and really biology in general from an evolutionary point of view. And particularly what we're talking about as far as molecular biology, these are things that are exactly the same. It doesn't matter what country you came from originally, or what kind of cellular organism you are. A lot of these things are really being used in extremely similar ways. All of us, and when I say us, I include the archaea and the bacteria and everything that's in my guts as well as all the other cells, are using the same genetic material, have a lot of very, very similar kinds of mechanisms that are going on. And so that's really the main point here. And yeah, we're over here. Molecularly, we're really kind of boring. Speaking, again, thinking about this from an evolutionary point of view, one of the really important things and concepts that I'm not sure if I got across completely is really this concept of conservation. If you find something which is the same across all of those different organisms, it's probably going to be really important and fundamental for all organisms. And that's particularly what's shown here in the green. And this is just the small subunit rDNA gene. This is the ribosomal DNA gene, which turns into ribosomal RNA. If you look at this section here in green, this is very well conserved, which probably means it's likely to be important in all organisms. On the other hand, this piece right next to it is not as well conserved. The sequences are not identical in all of these different and diverse organisms. So if you're interested in the differences between these different organisms, the different classes of organisms, I should say, this is the kind of place that you should be looking. So in general, conserved. More specifics, you're going to look in the less conserved region. And conserved just means similar sequence, which is really pretty amazing that that's how things happen. So as far as conservation is concerned, yes, this is completely conserved as far as we know, certainly for all cellular organisms. Central dogma, a couple of people have asked me questions about this. By the way, feel free to email me with questions up through Wednesday morning. I will do my best to answer them and post them on D2L discussion. This discussion group on D2L is a questions and answers where I post stuff. So central dogma, and this was brought up by a certain fellow by the name of Crick, and it's been followed quite a bit ever since, but one of the main things about that was in the 50s. And there are some things discovered in the 70s which they found was actually not completely correct. Like Crick wasn't completely correct with everything. So the Crick central dogma was DNA can replicate to DNA. And so DNA goes to DNA, and there's this whole circle. And then there's this linear process here, whereas DNA gets transcribed into RNA, and RNA gets translated into proteins. And so, of course, the big thing they found in the 1970s was reverse transcription, where RNA could go to DNA. And so the information, and this is really talking about information transfer here. It's the information in the DNA is stored as the information in the DNA, can be copied as information into RNA, translated into the information that you have in amino acids and proteins. And this process, the information you have in the amino acids, cannot be reverse translated to get you back this information, at least not in a biological system. And when we talk about genetic code, you'll see there's some clear issues why that is. On the other hand, there are a number of viruses, and in fact, even in our own human genome, quite a few proteins that can do this, take the RNA information and turn it into DNA. That was one of the questions on one of the previous exams. So this is the information transfer process. DNA to DNA, DNA to RNA, RNA to protein. Yes?
On one of the previous exams, there was a question about how Watson and Crick incorrectly hypothesized that there was reverse translation. And I was wondering why they would stop that.
Okay, so I think again, I don't know all the details on that particular question. I have to take a look at it. Best thing to do is to email it to me. But Watson and Crick's idea was really that it went from DNA to DNA, DNA to RNA, RNA to protein, not back in the opposite direction. And it was only when David Baltimore and Howard Temin found reverse transcriptase that they found reverse transcription to take place. Reverse translation, there's also some theoretical arguments on how that can't work as well. Yeah?
So the question
Yeah.
they're talking about is that the standard central dogma
Yeah.
and standard rotation, so that would be, they didn't believe in reverse transcription.
They didn't know about reverse transcription
So
at the time.
would be not included.
That would not be included in the standard, quote unquote. And that, again, this is as far as the exam is concerned. If there's something that comes up like that, so what the heck do you mean by standard? Put your hand up. That's why I'm here. And I'll try and answer that question. Okay, so this actually really leads into this slide that I asked if anybody had an idea why I put an elephant down in the corner. Anybody know why? No, it's not never forget, which is actually not a bad answer. No, so another famous molecular biologist, in fact, probably one of the most famous molecular biologists, François Jacob, is said to have said, si c'est vrai pour E. coli, c'est vrai pour l'éléphant. And basically, if it's true for E. coli, it's true for the elephant. And again, getting back to that whole idea of conservation, you know, molecular biology is really about these things that are similar. And so if it's true for E. coli, it's also going to be true for the elephant, which is true in some cases, but not in all. Again, we're biologists, so we know there always are exceptions to these things. Another thing that I wanted to maybe beat the dead elephant maybe here is talking about homology, paralogs, and orthologs. This is something that seems to be a difficult concept for a lot of people. It was for me before I went through it a couple times. Any sequence, which is going to be, you know, DNA, RNA, protein, which comes from a common ancestor, is a homolog. Now, usually you can tell that because they have similar sequences. But that's not always going to be the case. There could be a homolog that has a completely different sequence because they've mutated so much in the intervening time period. So just anything that has this common ancestor. Just because it has a similar sequence doesn't necessarily mean that it's a homolog either, because there should have been convergent evolution that has happened. But common ancestor means it's a homolog. So paralogs and orthologs are both homologs, because they've all come from a common ancestor. Now, the difference between the paralog and the ortholog is that orthologs happen because you have speciation, and in different species you have diversification. And here we'll just take the example of the G for globin. Beta globin in humans is an ortholog of the beta globin that you have in chimpanzees. So they're homologs, they're related to each other. They came from a common ancestor of human and chimpanzees, probably 10 million years ago, not that long ago. And then you also have the opposite, the flip side, which are the paralogs. And the paralogs are genes which have arisen from gene duplication in a single species and have then diverged in that single species. And a great example here is also alpha and beta globins. So the human alpha and beta globins are paralogs of each other, and they're both present in the same organism in that same genome. What happens with paralogs is they're usually going to have slightly different activities relative to each other, because why have two copies of the same gene if you don't need lots and lots of copies of that gene? In some cases you do, but in most cases you don't. Yeah?
Just to clarify, so paralogs are the ones that will usually have diverged in function to some degree, and orthologs have a similar function?
So the question here, but again, I'll try and restate this. So the question is, if paralogs diverged in terms of function, what about orthologs? So just to sort of paraphrase your question. Orthologs can have diverged in terms of their function, but in their different organisms, those different organisms may have different things that they need them to do, and have evolved in that way. But in many cases, they're going to have very, very similar functions. On the other hand, to get a paralog, you have to have gene duplication to take place. And so you've got two copies now of ancestrally the same gene. Now, if you've got two copies, one of those can mutate and do its thing and get a different function. And that potentially would be advantageous to that particular organism which has that. So in general, and again, these are ridiculous things to say in biology, you're going to have paralogs having at least slightly different functions, and in some cases, very, very different functions. And that leads to the gene families that I wasn't going to talk about here. But many of these gene families, like the protein kinase families, are massive and all have different activities. They're still protein kinases, but they've got all kinds of different substrates that they act on, for instance. Any questions on this whole ortholog, paralog, homolog, og? Yeah.
Don't sometimes, like when you have gene duplication, doesn't sometimes one of them get like silenced?
So the question. Yeah. So the question here is actually going to get back to that later on. You beat me to it. So the question is, what about some of these paralogs that are no longer functional? Let's say, sorry, paraphrase your question. Does that happen? And the answer is yes, it happens a ton. And when we talk about the human genome, I can now sort of skip ahead to that. There are almost as many of what are these called pseudogenes, so non-functional sequences, as there are normal protein genes in the human genome. So about 20,000 protein coding genes and about 20,000 pseudogenes. And so this is an extremely common occurrence, which makes perfect sense, because if you think about most kind of changes that are going to happen to a particular gene are going to be detrimental. It's only going to be very few of those. They're going to be actually something which is going to be positively selected upon. Okay, so paralogs, orthologs, etc. Why are these so useful? Among other things, the orthologs are really critical in different organisms, because this allows us to go back to that whole conservation thing that we talked about at the beginning. If it's an orthologous gene in different organisms, it's likely to have a pretty similar function. And so if you have a particular organism that has an orthologous gene to, say, humans, then it's often very useful to study that in that particular organism, because it's transparent, it's small, it has a quick generation time, smaller genome, good genetics. It actually mates with what you force it to do, as opposed to humans, which have a nasty tendency to mate with the people you don't want them to, at least as far as human genetics is concerned. So that's sort of the idea of these model organisms, is find orthologous genes, or for that matter, even orthologous processes, because ortholog doesn't necessarily have to be a gene, and then study those in that particular organism. And so, as I mentioned before, E. coli, c'est vrai pour le, actually I think it's the bacille de coli rather than E. coli, but true for E. coli, it's true for the elephant, it's true for us. And so studying some of the very basic mechanisms in E. coli, very small genome, great genetics. We've got basically know as much about this organism as we probably know about any cellular organism, but we're still finding new stuff out about it, which is still pretty amazing. My favorite model organism, Sulfolobus solfataricus for the archaea, because that's what we grow in our lab. And then a few of these eukaryotic ones, Saccharomyces cerevisiae, a lot of the stuff we're going to talk about as far as gene regulation, as far as cell cycle, as far as most of the fundamental molecular biology things, chromatin remodeling, etc., all come from studies of Saccharomyces cerevisiae. And the main reasons for that: small genome, relatively small number of genes, and really, really, really easy to work with. Grows on plates. You can make literally trillions of them. You can make them mate exactly the way you want them to. We'll talk about that mating system later on, etc. So a lot of the really basic biology in terms of eukaryotic biology comes from the study of yeast, and then a number of other different organisms, mostly because they got good genetics for working with them. Most of these other model organisms here have actually pretty similar numbers of genes as far as comparison to humans. Many of them very similar. A few of them have considerably smaller genomes. Ataliana for plants, and Drosophila, C. elegans, whereas humans and zebrafish actually have pretty similar sized genomes to ours. Switching gears, and again, we're going through 8 lectures really rather rapidly here. A couple things about chemistry. The main thing here has to do with all of the weak interactions, the weak bonds, all of the hydrogen bonds which are holding the 2 strands of DNA together, the actual stacking interactions which have more to do with van der Waals interactions, which are holding the base pairs together, hydrogen bonds that are also pushing nonpolar residues, amino acid residues together, because water just loves to form hydrogen bonds. All of these are relatively weak in the individual, very strong when you put all of them together. They're all additive, and you can break them apart, put them back together, break them, put them apart, put them back together. And that process really allows biology to take place. It's got to be dynamic. If it's not dynamic, then basically, as one of my colleagues always used to say, if you lose your battle with entropy, it's bad. Should we be expected to know that Stedman says a hydrogen bond is a delta G of 4 kilojoules per mole? No. That they are weak, and that the relative amounts, van der Waals are the weakest. Hydrogen bonds are stronger than that, and all of them are about 100 times less than a covalent bond. All of which are more so than the average thermal motions that you have, with the exception, again, of the van der Waals. So, but no, actual numbers I do not expect you to remember. I can't remember them. And actually, it depends a great deal exactly under what conditions you are looking. And so, if the numbers change, I can't really expect you to remember one versus another. Most of the covalent bonds, and these really strong bonds that I'm talking about, that we're going to talk about here, really break down into 2 reactions, which is, kind of, you can also think about one reaction being the reverse of the other. Condensation and hydrolysis. Adding water, removing water. 90+% of the reactions in biology have to do with condensation and hydrolysis. And condensation, loss of water, usually that's putting 2 monomers together to make a macromolecule, which is what we have down here. Breaking apart that macromolecule is a highly energetically favorable process. And to get any kind of polymerization to happen, fight against entropy, you have to have coupling between different reactions. And again, it's usually hydrolysis versus condensation reactions in order to get any kind of condensation, and again, usually forming covalent bonds here, to happen. What is a coupled reaction? A coupled reaction is just 2 reactions that are linked to each other, and basically conservation of energy. So if you have one energetically favorable reaction, as long as the reaction that it's coupled to has a slightly smaller overall amount of free energy, then one reaction can lead to the progression of a otherwise impossible reaction just from a thermodynamic point of view. And so it's this gear in the middle here. Large negative delta G allows something with a positive delta G to actually take place. And again, the vast majority of the reactions we're going to be talking about here has to do with polymerization. How does that happen in terms of nucleic acid polymerization? We'll talk about amino acid polymerization much later on in the course. Many cases, this has to do with some kind of so-called high energy intermediate. So it's a coupled reaction, and the coupled reaction actually is over here on the right-hand side, where you go from a short piece of, in this case, nucleotide, and extend that by one particular base. The coupled reaction here is the hydrolysis of these two phosphates. I think I've mentioned this before, but it bears repeating. These are called the alpha, beta, and gamma phosphates. Alpha, beta, gamma. Everyone had Greek in school? I didn't. I had Latin, but I never had Greek. So alpha, beta, and gamma phosphates. It's the cleavage between the alpha and beta phosphates which generates pyrophosphate, which is these two phosphates hooked together. And again, another high energy bond here, which gets hydrolyzed by the pyrophosphatase. And it's these coupled reactions which allow you to get polymerization. How do you get this in the first place? This is that high energy intermediate through the use of adenosine triphosphate makes these bonds. So hydrolysis of ATP allows you to make a triphosphate on your nucleotide. Chemistry's done. Woohoo! That was great. So more or less. So talked a lot about DNA. Also talked a bit about proteins. A couple of important things about proteins that I wanted to go over again. The first one is that just like nucleic acids, proteins have molecular biology polarity as well, which means they've got a start and a stop. And it's not 5' and 3' that you have in nucleic acids. It's N-terminus and C-terminus. Each of the individual monomers, which are amino acids, have an N-terminus and a C-terminus. The C-terminus gets hooked up to an N-terminus. C-terminus gets hooked to an N-terminus, and you have this polypeptide. That process has what's called the backbone. So this would sort of be the equivalent of the ribose, phospholipase backbone that you have in nucleic acids. Here it's the peptide backbone. Also here you have the so-called alpha carbon. That's the alpha carbon, which is right in the middle of all of these amino acids. That's where all the side chains are attached to it. And then the rest of it there. This process, all of these amino acids hooked up next to each other from an N to a C-terminus, is the primary protein structure. Primary protein structure is just the sequence of your amino acids. Those sequences of amino acids, through interactions with backbone atoms, and this is really important, it's the backbone atoms that are important for making the secondary structures. What kind of secondary structures do we have? Beta-plated sheets and the alpha helices, both of which come together because of hydrogen bonds that form between those backbone atoms that you have in each of the individual amino acids. What do these secondary structures do a really great job of? Sticking those side chains out, away from where the backbone is. Now, it's the interactions between those side chains, for the most part, which allows you to form the tertiary structure, so the secondary structure pieces coming together. And that happens through all of these really wonderful weak interactions, the van der Waals interactions, ionic interactions, the hydrogen bonding, etc. That will bring together all these secondary structures in order to give you a three-dimensional structure. And that three-dimensional structure usually is what's finally going to give you use function. So many cases you have the tertiary structures, each individual chain, and when people talk about a chain, that means it's got one N-terminus and one C-terminus. that then come together into a multimeric process, and that will be called your quaternary protein structure. Yes?
Are the tertiary structures only due to nonpolar and polar side chains interacting, or is there other types of interactions that build that?
Okay, so the question here is that tertiary structure just because of polar and nonpolar side chains interacting with each other. That's only part of it. So you also have charge-charge interactions. So what people will call salt bridges. So you've got between your charged amino acids, negatively charged versus positively charged, which I do expect you all to remember which ones are which. There aren't very many of them, so it's pretty easy. That happens also in terms of protein folding. You also have van der Waals interactions. You're also going to have your hydrogen bonds. So that as well as polar and nonpolar. So all of these things are interacting with each other. But probably the best example, thank you for the lead-in, of these polar and nonpolar interactions are the coiled coils, which most people call leucine zippers, which have to do with the side chains of alpha helices. One of the things that I think I forgot to mention, we talked about alpha helices before. If you go around 7 amino acids in your alpha helix, you end up on exactly the same side of that alpha helix. And so what that means, if you have a large nonpolar amino acid side chain, every 7 amino acids, it's all going to be sticking out on one side of that alpha helix. And that's exactly what you see here. Every 7 amino acids, there's a leucine, which is sticking out. Leucine is a large nonpolar amino acid. Those really love to interact with each other because the water likes to bind around the outside. And that gives you 2, in this case, alpha helices, which have bound to each other. When you talk about coiled coils, it's only going to be when you have an alpha helix, because that coil that is coiled around is your alpha helix. So 2 alpha helices coiled around each other. Every 7 amino acids, a large hydrophobic or nonpolar amino acid. Very often these are leucines that you'll see here. Often used for dimerization between 2 domains of a multimeric complex. Yes?
What do you mean by the 6 amino acids?
So, yes, it's, if you actually want to get into the very nitpicky details of the exact way, if you line up a vertical alpha helix, it's actually 3.6, and then it'll be 7.2. One of the things that you'll notice if you look at this coiled coil, particularly in this side view, they're not straight alpha helices. They're actually slightly bent. And that slightly bent actually brings them exactly into registers so that they do fit into each other. Yeah?
Just do a really quick definition of a domain.
Really quick definition of a domain. So, really quick definition of a domain is part of a protein that folds up into a specific tertiary structure. And that's, of course, specific tertiary structure. That is now going to have a particular function. So some proteins have a single domain. Most proteins have multiple domains. So they have multiple, but these are independent structures, and you take that piece of the protein separate from everything else, it will make that particular structure and have that particular function. On the other hand, these guys, just a single alpha helix that's got large hydrophobics every 7 amino acids is not going to just fold by itself. Okay, other questions here. So why do you have all these amazing things? Basically, it's all kind of window dressing, at least as far as most proteins and most enzymes are concerned. All of these hydrophobic interactions, polar, nonpolar, hydrogen bonds, van der Waals interactions, etc., are all about getting a few side chains into exactly the right place in order to bind to DNA in some cases, up here, bind to a specific substrate that you're interested in terms of enzymatic activity. But the vast majority of the amino acids in your protein, so you've got, going back to your domain question, most domains have 150 or so amino acids in them, 100 to 150. Probably that domain, 4 of those amino acids will be important for the actual function. And that would give you 96, which are involved in just getting the structure in the right place in order to have that really specific kind of interactions in an active site of some kind. Yeah? Would be a motif. So I've been avoiding talking about motifs this class because it just seems to confuse people more than anything else, which is maybe your question about domains. Motifs, since you asked, have to do with a sequence, just looking at the amino acids in the primary sequence, that will then tell you something about what happens with that. And so classic motif has to do with the leucine zipper. Every 7 amino acids is a leucine. That's probably going to be involved in some kind of coiled-coil interaction. But it's that primary sequence, you can see something in it. That's where your motif comes from. Again, maybe I should just not answer some questions sometimes. Sorry about that. Yeah, so what do we care about? Protein structures. A lot of these things, again, it's about protein structure and really how these different proteins are interacting with other things. And that really, if you're going to have a dynamic system, you need to somehow change structures. So the classic examples of changing structures are the kinases and the GTPases, or GTP binding proteins. Kinase phosphatase, put on a phosphate, clearly very different charge than you otherwise would have, and that will influence both the structure of the protein which gets modified and then anything else that it's interacting with. The kinases put the phosphates on, this side, and the phosphatases take them back off. G proteins, in terms of the very fundamental thinking about these things, it's a change in structure which then leads to a signal. But instead of a covalent modification, which is what happens with protein kinases, putting phosphates on, here it's hydrolysis of a tightly bound nucleotide. But still, it's the presence of that negative charge, or the absence of that negative charge, that leads to a change in the structure of this GTPase, GTP binding protein, and it's that change in structure which leads to different interactions, other things that are going on there. Different again because you don't have this covalent modification. It's the G protein itself which changes the structure. So it's got its own activity, bound to GTP, bound to GTP, bound to GDP, etc. This process happens spontaneously. It can be sped up by extra proteins, and once you've hydrolyzed this, the G protein can't become reactivated until there's another protein which can put in the new trinucleotide. But this process of turning itself off or hydrolyzing that phosphate happens just through the GTPs. This is the big difference between these guys. The other signaling proteins that we talked about are the ubiquitin ligases. Basically, all that I wanted to mention here is that I think I had a clicker question about did E3 have any kind of ubiquitin ligase activity? Why do you call it a ubiquitin ligase? Well, Dr. Singer said it's because it's got an E2 attached to it, but it turns out there are very few E3 proteins that actually will have ubiquitin associated with them. So probably wasn't a great clicker question. Should have asked him beforehand. But this process, you have, just like you have with the kinases, you have a covalent modification. That covalent modification here, instead of putting a phosphate on, is actually adding a small protein, ubiquitin, that gets transferred through these activated intermediates and eventually gets put onto a target protein. This targeting can lead to degradation, but it doesn't always have to lead to degradation. Okay, any questions on all of these things before we start moving over to the interesting stuff, namely the nucleic acids? That's what we've talked about so far. Yeah?
So if ubiquitin leads to degradation sometimes, what does it lead to other times?
Okay, so the...
a general function, or is it...
Yeah, so the question is here, if it's not leading to degradation, what's it doing? And the answer is, a whole bunch of different things. But the example that we have actually talked about so far has to do with modifications to histones. So those histone modifications that happen in the tails, those don't lead to histone degradation. Histone degradation would be probably a pretty bad idea. But what they do is change the interactions with other proteins. So your chromatin remodeling complexes, etc., which is exactly what's happening here. It's changing structure. How are you changing structure in this case? You're putting this big old, well, big protein. It's a small protein, but it's still protein. These 90-some amino acids you have in the ubiquitin. So it's that change. And so it's changing what else is interacting with. And it turns out that actually degradation is happening in exactly the same way. It's that polyubiquitin chain that leads to targeting to all the degradation machinery that we'll talk about later on in the class. So it's all about different protein-protein interactions, macromolecule interactions. And this class is, okay, other than the chemistry we talked about, it's all about macromolecule interactions. Okay, that other set of macromolecules, the nucleic acids. Wanted to mention this again here. Most people give Chase and Hershey credit for this, but I think much more credit should go to Avery and his colleagues for looking at this. Transforming principle. So what takes cells that look like this and turns them into cells that look like this, which they could do in the lab. And of course, these cells make the mice sick. These cells don't. You take these cells, mix them together with killed these cells, and these cells become those cells. So the transformation process. What's going on there? They were just trying to figure out what made this difference. What allowed these rough cells to become the smooth cells? And what they found was it's something that they couldn't get rid of with RNase, they couldn't get rid of with protease, but as soon as they treated it with DNase, it completely fell apart. You didn't get this activity anymore. And so that's really where the, I think, best evidence for DNA being the genetic material came from. Then, of course, about 10 years later, Watson and Crick came up with their model for what DNA looks like. Few of you have told me what at least one of the problems is with these models. One of the problems with these models is actually they got put together somehow so the strands are parallel to each other. How the heck they did that, I really don't know. I need to check the instructions for putting these models together. So clearly incorrect because they're parallel strands. You got a second after class, come down and take a look at that. That's not true. DNA and any kind of nucleotide that forms, relatively normal base pairs is going to be an anti-parallel state. So you've got 5' end at one end and a 3' end at the other, and these are opposite to each other. So if you've got a 5' end on this strand, it's a 3' end on this strand, and follows all the way down here. Through that process, you have, of course, base pairs in the middle, and those are hydrogen bonding with each other. Watson and Crick thought that these guys only had 2 rather than 3. Yeah?
So when Watson and Crick thought that DNA replication was continuous,
Mm-hmm.
right? So they expected there to be a 3 to 5 polymerase?
Okay, that's getting a little ahead of ourselves, but Watson and Crick expected that replication was going to be continuous, and there's a little line in their paper, which is probably one of the most famous lines in molecular biology that I won't quote because it might show up on an exam, talking about how replication could be taking place. And what they thought is the 2 strands came apart and they were both replicated in a continuous fashion, which makes sense if you, think about it from a nice symmetrical model system. These guys are modelers. Many of them came out of physics originally. So nice, easy, of course, biology isn't like that. So one of the things that came out of Watson and Crick's model was that you had not only these base pairs that were forming in the middle, but another problem with this model is that all the base pairs here are right exactly in the middle, which is not the case. If there were, there wouldn't be nice major and minor grooves relative to each other. They're offset. And that gives you a major groove in your DNA and a minor groove in your your DNA. Why are those important? It's because all of the information that you need to be able to read what the base pairs are is present in the major groove of DNA. So if you want to have specific interactions that are happening with a double-stranded DNA, so you don't have to pull the two strands apart to get the information out. You can get all the information you need just by looking in the major groove of DNA. And that information is the chemical information that you have with hydrogen bonds that could serve as hydrogen bond donors here, atoms that can serve as hydrogen bond acceptors, methyl groups. Again, one really good way to tell where the major groove is on your DNA is just to look for where the methyl group is on all your thymidines. Here, it's a black ball, so it's really easy to find in the major groove of DNA. One of the really fascinating things about DNA that has allowed us to do really amazing things in terms of genetic engineering, general molecular biology, is that you can pull the two DNA strands apart and they come back together, and they come back together very specifically where the base pairs are actually forming. So as long as you don't have highly repeated DNA, which unfortunately our genome actually has a whole bunch of repeats in it, so it's actually a little harder to do this, pull the two strands apart, they will come back together, and they'll come back together in exactly the same place. And that transition between being together and coming apart is a relatively sharp transition. So once you get the two strands that come apart, they'll very often come apart completely very rapidly. And that's shown here, not so much in terms of time, but just in terms of temperatures. There's a very specific temperature right here, the melting temperature, at which you have half of your DNA together, half of the DNA apart. You can tell that because the absorbance of your DNA in the ultraviolet is actually relatively low as double-stranded DNA and higher as single-stranded DNA. So you just look at that particular wavelength. It has to do with the absorbance of the individual base pairs. We won't get into that. If you're really interested, take spectroscopy and chemistry. But pulling those two strands apart lowers the absorbance of your DNA and happens at a very specific kind of temperature. Yeah?
Does the percent of CG composition in your DNA alter the melting point at all?
So the question is, does the percent of GC alter that melting temperature at all? And the answer is absolutely yes. So if you have higher percent GC in your DNA, this melting temperature is going to be higher. A lower percent GC or higher percent AT, it's going to be a lower melting temperature. And that's in fact one way that people try and figure out whether the new DNA you found actually has high GC or low GC. You put it in a spectrophotometer and heat it up and see what temperature it comes apart. So pretty straightforward way to figure that out. Okay, so we actually talked about this already, talking about pseudogenes, these paralogs that have formed in the human genome, which are actually then no longer functional. A couple of other things about the human genome. Yes, about 3 billion base pairs in length, around 20,000 genes. And when I say these are the numbers that I mention, those numbers are probably going to be important, around 20,000 genes, that it's 21,000 genes, or that the largest exon is 17,106. No, that I don't expect you to remember. But at about 20,000 genes, and the important thing with those about 20,000 protein-coding genes, that's really similar to what you see in C. elegans. It's really similar to what you see in Drosophila. So there are very similar numbers of protein-coding genes in many, many, many of these different organisms. So that's the main sort of take-home message that I wanted to get from here. The only other one is the double asterisks, which we have down here. I can get my pointer to work. Number of non-coding RNA genes. The double asterisk here basically means question mark, question mark, question mark, question mark. We really have very little idea how many non-coding RNA genes there are. In fact, since this textbook was published, this is almost doubled. in terms of the number of non-coding RNA genes that are present in the human genome. And there may be way more. So this is the really big open question. Another number that I do expect you to remember, because I kind of talk about it a lot, is this 1.5% of the human genome which codes for non-repetitive proteins. Turns out there are lots of repetitive proteins that we'll talk about well after the midterm. But what makes us human in terms of protein coding is that 1.5%. So you've got a big genome, actually 6 billion, 6.5 billion base pairs that all need to be scrunched down into a nucleus, a couple of microns in diameter. How do you do that? Stretch out all the DNA, it's taller than me. So how that happens, of course, it's chromatin, DNA plus proteins. The first step of that is nucleosome formation. Nucleosomes, of course, are made of histones. All of the core histones, when I say core histones, it's everything but histone H1, H2A, H2B, H3, and H4. Those are the core histones. All have a very similar structure. And it's one long alpha helix with two smaller alpha helices crossed across them. That's the core of the histone fold. And then all of the core histones have tails that hang off, either N-terminal or C-terminal, to that histone fold. Because of the histone folds, those all associate with each other. And as we talked about before, if you look at how nucleosomes assemble, and this is a reminder to go back and look at that video in terms of looking at the assembly, that is a very good scientific interpretation of what's going on there. First, you have histone H3 and H4, which interact with each other, the blues and the reds, the blues and the greens on here, and then you have H2A and H2B, which associate. That's important for a couple of different reasons. The main one is that when you're exchanging the individual histones, then it's easier to exchange H2A and H2B rather than H3 and H4. That's just an overall issue, but not always. Sometimes you can also have alternative histones. All those alternative histones that are H3, H4, H2A, H2B, those all still have the histone fold so that they can assemble inside the nucleosome. Nucleosomes are great. They do a nice job of compacting your DNA, but they immediately bring up the problem of how do you compact the DNA but still have it accessible to all the other things that you want to interact with your DNA, all the other proteins, etc. So you have to have a way of moving histones around. And the main way that that happens is through these chromatin remodeling complexes that we looked at last time. And actually, I think I told you it was 2 weeks before that the paper was published that we talked about. Actually, it was published on Friday, and I talked about it on Monday. So it's actually only 3 days old rather than 2 weeks old. So what seems to happen with these ATP-dependent chromatin remodeling complexes is they basically jiggle where all of those core histones are relative to the DNA, and that allows the DNA to move relative to the nucleosome complex. So that process either can just move the DNA along, or it can cause dissociation of the nucleosomes, either in terms of getting exchange, and usually, again, this is H2A, H2B, which will be exchanged. In some cases, however, you can completely disrupt the nucleosome and have a completely new one put on. Usually it's this top process, sometimes this bottom process. Couple of things that are important here. Also, it's all ATP dependent. So the nucleosome structure in and of itself is really pretty stable. Lots of hydrogen bonds, hydrophobic interactions, charge-charge interactions, etc. So in order to change that nucleosome structure, you do need to have some ATP input. However, when you do have histones that are already assembled with your DNA, you can still change what's going on with each of those individual histones because of these tails that are hanging out, both N and C terminal. Again, the best studied of those is probably histone H3, and that's here just the sort of cartoon version of the N-terminus of histone H3. You can see where it hangs out here in the structure, outside of the nucleosome, so that you can either interact with other nucleosomes, sort of packing your 30 nanometer filament, but also with other proteins that are coming in. And so it's really this, changing interactions with other proteins. How are you changing those interactions with other proteins? Making modifications. So this is where you get ubiquitination that happens, which is not listed as being one of the modifications here. Some textbooks actually include ubiquitination here. There's another sort of peptide called SUMO, small ubiquitin-like modifier, that also modifies these histones. But it's still the same basic idea. Make a chemical change so that you have differences in terms of what other kinds of proteins can interact there. And so those can be other histone methyltransferases. We talked a lot about the reader-writer complexes. So the readers are going to be the ones that recognize those modified histones. Individual domains, get back to your domain question, very often it's a domain that's going to interact with one of those particular modifications, which is there. Multiple domains are going to come together with these reader complexes, which are very often connected to writer complexes, which lead to maintenance of this particular state. So this is one of those slides that I think I did. Oh, sorry.
A question on one of the old exams
Mm-hmm.
was concerning the histone modification or the remodeling complex
Mm-hmm.
and ATP-dependent covalent modification.
Mm-hmm.
Is that going to be referencing just the tail modifications or the swapping of the dimers?
Okay, I'm not sure I completely understand your question because I don't remember all the questions on my old exams. Go figure. So I think what you're driving at, right, has to do with covalent versus non-covalent changes. The chromatin remodeling complexes don't make those kinds of covalent modifications to histones. They're just moving the histones around. So that ATP hydrolysis is not actually forming or breaking any kind of covalent bond. On the other hand, the histone code writer complexes, the histone methyltransferases, the acetyltransferases, those are making changes. Those are going to make covalent changes. Yeah?
But that's on the tails, right?
On the tails, yes.
Right, okay. Because there's something in the book that talks about side chains within the core.
Yes, so, okay. Good reminder here. There will be things that are in the book that are not in my lecture. I wouldn't concentrate too much on those. But as always, and you could, every lecture you could raise your hand and say, Dr. Stedman, isn't this ridiculously oversimplified? And I'd say, yes, of course it is. And that's getting back to that point. There are actually core histone modifications that are inside that get modified. How the heck that happens and what it does, I have no idea. So that's one of the reasons we don't talk about it. Yes?
People want to know generally what phosphorylation or methylation or acetylation does on a specific, like on a lysine or on a tyrosine or something.
Right. So the question here has to do, actually kind of similar question we had here about knowing the actual numbers of your kilojoules, et cetera, per mole. Do I expect you to know what a very specific modification to a histone side chain has to do with expression versus heterochromatin formation versus euchromatin formation? The answer to that question is nobody really knows, so I don't think it's reasonable that I expect you to know.
That's comfortable.
So lots of variations and things that happen and lots of differences which happen there. Okay, so if we don't get to the end here, that's perfectly fine because hopefully it's in your short-term memory anyway because we just talked about DNA replication. If people have other questions, they can get back to me. So we'll finish up in the last 10 minutes here and I'll try not to talk too fast through them. Couple of things that I thought I did a really, again, crummy job of explaining. One of those were these lampbrush chromosomes. What's going on with the lampbrush chromosome? Lampbrush chromosomes are what you see in amphibian oocytes, which are basically frog eggs, because that's what 90% of people are looking at. And when you just look at these chromosomes, which are actually a whole bunch of chromosomes lined up next to each other, then what you see are most of the DNA is actually down here at the bottom. The vast majority of DNA is down here. There are a few of these loop structures that stick off of, again, the vast majority of DNA that's down here. And people have done quite a lot of experiments, and they've shown that it's these loops here where the DNA is being transcribed into RNA, and that RNA is eventually being made into protein. You'll see there are actually a relatively large number of loops here. If you think about an oocyte, particularly a fertilized oocyte, it's cranking out protein because it's got to undergo development, all of these other kinds of things that are happening. So if you want to have really transcriptionally active, fertilized oocytes are really a good place to go. And what we think we know now is in a normal eukaryotic nucleus is most of the DNA looks like this too, only you don't have many, many copies of them, so you can't see it anywhere near as well. So these loop structures, which have genes on them that are being expressed, they're separated by barrier proteins. If you just look at where the barrier proteins are, it turns out they're down here at the bottom relative to all of these different chromosomal domains. We also know if we look at polytene chromosomes, which are salivary glands of various different insects, again, multiple chromosomes all lined up with each other. They're all lined up with each other, and they're actually not active. You can see these nice bands here. In order to become active, you basically don't see any bands. And what that tells you is most of that DNA is in some kind of heterochromatin. It's compacted. In order to get it be transcribed, it's got to become euchromatin, which is less compacted. And we talked about the whole process, how that happens, etc. Before. So this we've probably already covered. Again, the main thing here is you go 1.5, the really minuscule part, which is protein coding here. Lots and lots of repeated sequences that are present in the genome. And of course, the important bit here are, of course, all the virus sequences which are there. So how do you replicate this genome? It's the DNA polymerases. DNA polymerase 1 in particular, first one study, but it turns out that all of them are like this. They're not like Watson and Crick. You don't have the opposite direction in terms of polymerase. All of them start at a 3' OH and use a template and extend that 3' OH through the hydrolysis of the beta and gamma phosphates that we already talked about. So to get DNA polymerization to happen, you have to have a primer, you have to have a template, and you need these nucleotide triphosphates. So you need all three of those things. This means, obviously, that you've got a problem in terms of starting. How do you start? You have a primase. It's that primase, which actually is an RNA polymerase, which puts down a couple of nucleotides of RNA that then have a 3' OH, which can get extended. I mentioned this before. It was kind of out of order in the lecture when I talked about it the first time. When you have any particular piece of DNA and any origin that it starts from, you're going to start out at one particular position, and then you're going to have two replication forks that are going to be moving away from each other. So there's a replication fork going that direction. There's a replication fork going this direction. At each of those replication forks, you're going to have two templates because they've been pulled apart. It's the DNA helicase which pulls those two apart. One of those can be polymerized from 5' to 3' by everyone's favorite polymerase. But the other one has to be put together in a discontinuous fashion because it's got to go basically in the incorrect direction. And the way that this gets solved is it flips around. You've got the whole trombone model. Again, I recognize taking a look again at the animation as far as that's concerned. Here you have each of these fragments, which has been started by an RNA primer and then extended towards the end. Talked about the sliding clamp before. Basically, this is the processivity factor and the tool belt. It holds the DNA polymerase, again, by protein-protein interactions. Again, how many times do I talk about protein-protein interactions in any given lecture? Holding that polymerase on so it doesn't fall off of the DNA as you're undergoing replication, but it also provides interaction sites for all of the other proteins which you need, particularly dealing with primers. Yeah? You're going to have one on the leading and the lagging strand because the polymerase itself has to be held on. You'll have many more of the sliding clamps on the lagging strand because once it gets to the end of the Okazaki fragment, then that sliding clamp stays there and actually helps get all of the stuff there that you need to get rid of primers. So we already talked about RNase H. DNA polymerase always promise that I don't make changes from lectures to this review. This is where I actually did make a change. And that is that DNA polymerase 1 has not only a normal polymerization activity, 3' to 5', not only a 5' to 3' exonuclease activity, which is the proofreading activity backing up, it also has a 3' to 5' exonuclease. So DNA polymerase 1 can back up and chew up nucleotides, but can also go forward and chew up nucleotides in front of it. So what's our normal polymerization direction? 5' to 3'. 5' to 3' exonuclease. What the heck is that for? It's for getting rid of primers. So it turns out that just like you have the flap endonuclease in eukaryotic systems for helping to get rid of primers, you also have DNA polymerase 1. Now, DNA polymerase 1 is only present in bacteria. It's not present in eukaryotic systems. But what it can do is be extending from the 3' OH, extend through here, where your RNA used to be, your RNA primer, and actually keep going. It will chew off individual nucleotides here and replace them behind it. So DNA polymerase 1 you can also use to get rid of primers. And it probably actually is used for that in E. coli to a great extent. What do you think about DNA polymerase 1 relative to interactions with the sliding clamp? Would you want DNA polymerase 1 to be a processive polymerase? Heck no, because it's got this counterproductive exonuclease activity. So once it gets rid of primers, you want it to fall off. So it turns out that DNA polymerase 1 is not processive and it doesn't associate with the sliding clamp. So I'm just going to stop here with this slide, which is actually almost my last one, and sort of review what we've talked about in terms of the replication process. DNA helicase, and this was the last part I would talk about, origins. The key to having an origin start is getting that helicase onto the strands. Once you've pulled the two strands apart, which is what the helicase does, then everything else is going to happen. You'll have the primase, which you'll put down your RNA primer. That gets extended by the replicative DNA polymerase, 3 in E. coli, delta in eukaryotic systems. That will extend through to the end of the Okazaki fragment. That gets degraded by DNA polymerase 1, RNase H, etc. You also will have had a primer way back here when you started. Still you got that sliding clamp, the question you had about the polymerase here. Always a sliding clamp. Holding on the polymerase, putting on the sliding clamp. You have the clamp loader protecting your DNA. You have your single-stranded DNA binding protein. Any more questions? I know a couple people have to leave. Please send them to me or come up to me after class. Good luck on Wednesday.